Hello everyone, welcome to Biology with Medmelo. And now we are going to discuss question number 9 of genetics. That is the concept of a gene, classification of genes, comparison of structure and expression of genes in prokaryotes and eukaryotes. Hence, basically, this question is based on the whole concept of gene, determining what is a gene, its types, and how it can be formed or regulate the function in prokaryotes and eukaryotes. So let's start. First of all, gene is the functional unit of genome. This is the major point. Every hereditary characters are made in some genomes. That's functional unit is gene. That is, every character would have some kind of gene, or it is formulated or saved in form of gene. And it's a region of DNA that controls hereditary characteristics. That is a part of the DNA that holds those hereditary characteristic, or that genetic information that is to be coded to a specific single protein. Now let us know the types of gene. Basically, there are five types of gene, and their function is basically determined by their name. Suppose we have first part, that is structural gene. Structural gene has the word structure in between it. That means it will be coding for a protein that will be forming what? The structure of something. That is, it codes for structure forming proteins. Those proteins which form some sort of structures in the cells. Secondly, we have regulatory gene. It has the word regulate in between. That is, it codes for a regulatory protein, and that regulatory protein's name is given to us, that is repressor protein. That is usually function in the transcription. Then we have the gene of histone proteins. That is, it will code for histone protein. Then gene of tRNA and rRNA means that they code for the protein whose end products are tRNA and RR. Okay, so let's find an easy way to memorize all these genes. Five types of genes determined by their name. Structural gene codes for structure forming protein. Regulatory gene codes for regulatory protein. Histone protein gene codes for histone protein. And the gene of tRNA and rRNA, their products are tRNA and rRNA. Okay, easy peasy. Now let's talk about the gene expression in prokaryotes and eukaryotes. We all know that prokaryotes and eukaryotes are two different types of cell bodies where cells are categorized on the basis of their type of nucleus. Prokaryotes don't have a true nucleus, right? As we have studied earlier, their true nucleus is absent, while eukaryotes have a true nucleus, which means that in prokaryotes, the gene distribution is uneven, while over here, the gene is distributed in the nucleus evenly. That means the prokaryotes have simple gene expression, while the eukaryotes have more complex gene expression. So let's talk about prokaryotes first. First of all, since there is no true nucleus and the gene's distribution is uneven, that means the gene expression takes place in cytoplasm. Secondly, the sequences of the nucleotides codes for amino acid to form into a protein, in which this procedure occur in two stages. And these two stages are, first of all, transcription and translation. Now let's move on to the eukaryotes. Eukaryotes, in the eukaryotes, since the process is very complex, the gene is divided to two sequences, coding sequences and non-coding sequences, due to which this gene expression would occur in three stages. And these three stages are what? Transcription, processing, and then translation. Also, during this transcription process of eukaryotes, the enzyme RNA polymerase will copy to RNA, DNA would divide to two sequences, exon, the coding one, and intron, the non-coding one. And the resulting RNA would be known as messenger RNA. Hence, the end product of the eukaryote is messenger RNA. So that was all about the question. I hope you understood it well and it will be easy for you to memorize it well. If you have any doubts, please let me know in the comment box. Thank you.